The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 21st episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the weekly bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian, and I've been in the flow cytometry space for over 13 years.
Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, with additional locations in Europe and a partner location in Australia, we're both here in Philadelphia, and Jeremy, our producer, is in Missouri. So we're truly all over the map. We're excited to have you listening to our 21st episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you'll likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources and a focus on a topic of our choosing before discussing any feedback we receive from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're really excited to have you here and looking forward to a fun episode today. To kick us off for episode 21, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. We've got a high-impact agenda today. Not packed, but very impactful, I think.
Now, is there an impact factor for that? Have we established one? What's our number here? Like 0.5? Are we higher than that?
I think our baseline impact factor for this one, again, because it is high impact, I would put the impact factor here as 7.
That's actually really good. I mean, the paper that we're going through today is an impact factor of 7. So were you using that or?
No, I'm just guessing on a 0 out of 10 scale. How impactful is this episode versus others? And I think we're upper middle, definitely upper middle. And the reason for this in part is because of the medical research news. And I'm always a huge fan and follower of anything related to gene therapy. And specifically, this one was very cool. So our first medical research news story is a very successful gene therapy that was carried out in Philadelphia. And the reason that this one was so cool is that it's what's called an N of 1 treatment. So that means that usually when you develop a drug, you're looking for relatively large populations to use that drug on. That way you can really benefit from economies of scale there. But sometimes diseases can be very unique to an individual, especially in the case of genetic diseases.
Now, it's interesting. So as I'm thinking about this, it's almost like gene therapy is going in the reverse of cell therapy, at least in this aspect, right? Because what are we trying to do with cell therapy? We're trying to go from customized autologous therapies to off-the-shelf allogenic therapies. And here we're going from gene therapies that are broadly applicable to an N of 1 that's really crafted towards the mutations that this one patient had, which, I mean, it's fantastic. And, you know, it talks to the difference of proteins versus DNA and, you know, recognition and all of that kind of stuff. But taking a step back, using technology called CRISPR, right? And I would imagine a large portion of our audience knows what that is. But for those that aren't familiar, CRISPR is spelled CRISPR. So it's clustered regularly interspaced short palindromic repeats. And I'm not going to try to say that really fast.
Well done.
And basically what it is a defense mechanism for prokaryotic organisms, right? Essentially against viruses. And the Nobel Prize was awarded in 2020 specifically for this. And a cool fact about that was there was the first Nobel Prize awarded to 2 women. And so really cool there. Essentially, it's very specific to a target, right? It can go into the cell, cut the DNA, and either remove a gene or implant a sequence or whatever to ameliorate a disease state. And it's very effective in that way. Now, there is some concern of off-target effects. It's still very small, but possible. And so one of the things that is necessary is sequencing to ensure that the gene was inserted or the change was inserted in the region that was needed. But then to your point, this is very customized for this patient. And it was interesting. They're talking about a 7-month-old, right? And so they said it went from sequencing to therapy in 6 months. So essentially, when they probably found out, right, a month old or something like that, it's just amazingly quick to be able to get to this point.
That's it. You think about a regular drug development timeline of being decades, right? If you get one through in 5 to 10 years, that means that basically a lot of the development work's already been done. Whereas from scratch, like from understanding an indication, usually that takes, at least 10, if not 20 years to really understand the mechanisms behind it and then target a drug. So can't emphasize enough the speed of this and how quickly it went.
Yeah. And some of the interesting things, generally, gene therapies tend to work better on younger patients than older. My nephew had gone through a gene therapy trial, and, for him, it was amazingly successful. And essentially, when he was born, we were told he had 29 months to live, right? And he just celebrated his 8th birthday. So speaking to the incredible power that something like this has, to be able to see him go from when he was born, he couldn't move, right? He was completely bedridden, breathing tube, feeding tube, all of that stuff. And now he can walk. He has played stuff like t-ball and whatnot. Muscle function isn't 100% of a child at his age, but still able to do the things on his own. He can eat on his own, things like that. And so it just changed immensely what would have been the path that he had ahead of him.
And it's wild, right? And so you mentioned CRISPR, and as you say, most people are likely familiar with it in our audience. And just really briefly, it is a protein, and then it binds to a piece of RNA called the guide sequence.
Mm-hmm.
And this guide sequence is what directs the nuclease activity that is doing the cutting and potentially insertion and things like that. So the promise of it is that this protein is always the same. It's considered like a pair of scissors. It doesn't matter what it's trying to cut. It is always just going to cut things.
Mm-hmm.
And then you can just change out this guide sequence, and that can rapidly target it to a new disease. And that's why this is so exciting for an N of 1 trial, and why we can get to smaller populations. Because if you validate basically your mechanism of delivery of the protein and mechanism of delivery of a oligonucleotide, then you can, in theory, change out the sequence of that oligonucleotide and have a very rapidly iterating treatment for different types of therapies there.
Mm-hmm.
And that's what they leverage in this case. And they've got a lineup of 12 other diseases that they're ready to go as soon as they see another case of it, that they can try out the same approach on.
Now, I am curious about your history with this, because you've mentioned, I think, a couple of times specifically that you're keenly interested in gene therapy. And so I'm going to give you the opportunity to talk about that.
Sure. I mean, in my grad school work, I worked quite a bit with CRISPR. We were primarily looking to detect off-target sequences and understand what could predict like where it's more likely to get an off-target mutation. That way we could, as we're trying to design therapies, design the therapies to target a place that is less likely to have off-target effects, right? And just improve the overall safety profile of these things. So definitely a connection in grad school there, but then also, like you, I have a nephew that has a pretty severe genetic issue, and is unfortunately not a candidate for this type of gene therapy because right now the cause of that genetic issue is too complex. Like we're still limited to pretty specific mutations and like one or two mutations with
Yeah.
CRISPR, like literally one in most cases, right? Whereas this specific mutation is a cluster of potentially as many as seven different mutations that can happen.
Mhm.
So when I get excited about gene therapies, I'm thinking, yes, that's exciting for the individuals involved, but every step that we get is a step closer to being able to do these more complex treatments that you could potentially address some of these 7X mutations and things like that.
And we've come a long way from the one gene, one disease modality that people, basically the pharmaceutical industry was on for quite some time. I do want to also point out, you mentioned about CRISPR, like that's the whole point of this article. There are other mechanisms for gene therapy, right? Lentiviral delivery, AAV. Some of the issues with those could be liver toxicity, and there are some rejection, right, for repeat tries, and so these are things that the industry is looking to prevent. And this is where something like a CRISPR has the ability to step in as well. So lots of options out there, and lots of tools that are being evaluated.
Absolutely. But, just to come back to the initial story here, again, just a wonderful outcome for the patients and just a huge success of the medical community to really bring this from basic research. You mentioned the Nobel Prize was awarded in 2020. This is one of the fastest routes from discovery to Nobel Prize in scientific history, because it was just recognized that this is a very high impact discovery scientifically. And then to go from 2020, the Nobel Prize being awarded, to 2025, therapy being developed in 6 months, like this is unheard of in terms of the speed that it's moving from basic science to wide applicability to the clinic. So definitely a huge impact there.
Yeah, and I think to your point, wasn't it about 10 years, like from the discovery and identification and of the features and all of that to the Nobel Prize? Really, I mean, how many times you see a Nobel Prize awarded posthumously because it had been decades and decades since discovery.
That's it. It's very difficult to know in advance like what's really going to be impactful. But when this one came out, it was just immediately clear that this is going to change. First of all, it's changed the way that science is done in academic institutions, but it's going to continue to change the way that we can practice medicine in the future as well. So
Yeah.
excited about more to come there.
Mm-hmm.
But you mentioned, traditional and legacy science, which still has quite a bit to give us. One of those is, of course, vaccines. So in terms of other medical research news, there's been a lot in the month of May related to vaccine regulation and guidance changes overall. So I did want to at least speak to that because it's definitely very prominent in the news cycles recently as well.
It is, and I think this is one of those ones. As a scientist, it's you really have to sit and see where it takes us, but there is a lot of hesitancy here, right? Vaccines have demonstrated immense potential and immense results. Let's not even call it potential, in eradicating diseases that had killed people, caused horrible effects, and unfortunately, now we're seeing some of those diseases coming back. And part of it is just because there hasn't been as much funding for vaccine education within the government. Here, what we're seeing is now, was it RFK removing all 17 panelists from the vaccine advisory panel? And so the article points out that it was done because of a claim of conflict of interest. However, it was based on a report from 2009, and really doesn't support that finding, at least from the various articles that we've seen here. And so now putting in a new panel. And I think, first off, as a scientist, right, having counter arguments is not unusual, right? That the science is built on trying to get to the best answer. It's not just saying you automatically have to believe in something. And so to be skeptical of something is one thing. Some of the discussion around the new panelists is more of an right? An anti bent here. And it's difficult to be wholesale anti on something that has been so beneficial for humanity. And it kind of, you worry as a scientist that it's going to detract from the progress that has been made.
Sure. And, actually the main article that I was trying to reference here is actually the NEJM article. That was actually the FDA's stance on vaccines. And it was interesting that they came out so close, right? Like the NEJM, so the New England Journal of Medicine, their article came out on the 11th of June to talk about what the expectations are for vaccine development moving forward.
Yeah.
And, towards your point about science, it was at least evidence driven, right? It said, this is what we expect out of trials. This is the benefit that we expect to see. We can talk about, are the thresholds set at the wrong level, but it is a data driven process of moving that. Whereas you're absolutely right that, I thought it was important to contrast that to, some of the other news stories that are coming out that say that essentially this is moving without the backing of science, and in some cases specifically contrary to it. So definitely a concerning thing. It's clear that vaccines are an overall net positive for society. Like they've played a huge role in just completely eradicating diseases that we've forgotten are so bad and so challenging to human society. So hoping that we can maintain a data-driven discourse overall, and that it's clear to all U.S. institutions that we need to maintain vaccination. Important for flow cytometry because we overlap so much with immunology, and the ability of our immune cells to recognize pathogens and attack them is really so core. So that's where it sort of ties into our flow cytometry podcast here. But definitely concerning news from the vaccine space, and hoping that, again, we really rely on this data-driven process to move us forward and keep us on the right track with vaccine and the wide-scale adoption of vaccines overall.
Yeah, I certainly agree. Hopefully we can maintain herd immunity and continue to keep these diseases that have been in check at bay. I think one of the hardest things for the public is that a lot of these diseases, you hadn't seen the horrible effects of them during your lifetime for the most part, because of the fact that it had been eradicated by vaccines. And so now you start seeing those small pockets that develop, and, it's the next door neighbor. It's small, right? But you don't want it to get to the point where it's bigger. And so it's really important that the government leads in demonstrating the science, right? What does the data say? Just like you were saying, and just really wanted to reiterate your point, because I think it's very important that we have a government that leads us by facts and data and that sort of information.
Yeah. Yeah, so we'll see what happens, but again, hoping for reasonable outcomes there. In terms of other people hoping for reasonable outcomes, I'm sure that BMS is hoping for a reasonable outcome of their new partnership with BioNTech.
Yeah, this is a big one. Looking to develop BNT 327. So we're looking at a bispecific. And it's interesting seeing, I wouldn't say that bispecifics had gone away, right? Certainly bispecifics were very hot. And then we saw the advent of cell therapy and gene therapy. But it seems like the market really is moving back towards bispecifics in a strong way. And this is one of those indications. So $11.1 billion centered on developing this anti-PD-L1 VEGF-A bispecific designed for cancer treatment, right? Now, the companies can each evaluate it for other indications independently, which I thought was pretty interesting. There would be milestones as they go through for funding here, and so important as to the value of this. And yeah, it looks like the drug is currently in over 20 clinical trials, at least 2 phase 3 studies in breast cancer. Sorry, 2 phase 3 studies in lung and also breast cancer. So definitely very strategically important in bulking up their portfolio, BMS's portfolio here.
You're right that it is interesting that bispecifics have become such a hot topic again recently, right? Like I think understanding that a lot of these cancers that we're trying to attack have complex mechanisms of action, or have difficult to target sort of immune landscapes, may be driving this, right? That we figured out that a monotherapy just will not do it. And even if you have 2 therapies delivered in tandem, it might not do it. But if you have 2 targeting moieties, then it's going to be able to basically still maintain efficacy despite any types of mutations in the cancer or changes in the cancer that would ordinarily potentially mask it or make it less effective. So very, very interesting from that perspective. The other big thing, just from a financial perspective, usually when you see these deals, they're very heavy on what investors call bio bucks, right? So it'll be, oh, here's this $2 billion deal for something. And then you look into the details and it's like a $20 million upfront payment, and the rest is just dependent on things going through, right? So there's just a lot of contingencies. This one was different in that it had a pretty significant upfront payment of $1.5 billion. So it just shows you that, when an asset has been fully de-risked, what the real value of it is. And then the rest of it does come in later, of course, but it's a much smaller proportion than a lot of these deals otherwise are.
Yeah, and the big pharma players always looking to one up one another and pull in these assets, right? And you think about these numbers, how much they're paying for it. If I read correctly, BioNTech had actually purchased this previously for much less, and now it's getting purchased themselves for significant increased investment. So, always keeping out an eye for an investment that you can turn over kind of reminds me of like flipping a house, right?
That's it, flipping a drug asset. Yeah. Well, we'll have to have someone on from a financial engineering perspective to talk about how that works in general. Maybe find somebody from a Vant and see if they're willing to talk about it. But yeah, I mean, it's definitely a big deal. Exciting for patients and for just treatment in general. So as we say, like the more treatments we get, the more different options there are. So that ideally, of the thousands of different types of cancers, if and when we ever get one, then our specific one will be covered in terms of one of the dozens of therapies that are out there. So hoping that goes well.
Now, in relation to the paper that we have, I think the comment that both you and I were just driving over was that there has been this shift. You're starting to see more of the bispecifics coming along, and that we have, I wouldn't say cell therapy is on the back burner, certainly. I mean, there's lots of companies that are doing it in earnest, but it just feels like the heat has been turned up right now. And so what are your thoughts there?
Yeah, I mean, a lot of pharma companies are coming up on patent cliffs, right? So they need to replenish their pipelines with differentiated treatments that are going to basically tide them through the next 10 or 20 years until they hit the next patent cliff, and then they'll need to go through another burst. So, if you look back 10 to 15 years ago, that's exactly what happened with big pharma, and that's why there was a wave of really mega mergers as they needed to replenish their pipeline with ready to go assets. Because especially with that last round of patent cliffs, most of the investment community thought that big pharma actually waited too long. So because they waited too long, they had to find drug assets that were completely ready to commercialize Whereas now they can be a little bit more selective and purchase items that have not been fully de-risked,
Hmm.
but they can get that at much cheaper prices. So that's likely what's driving a lot of the push right now for M&A activity.
Yeah, and the prices, that was something that I definitely had in mind, the cell therapies being much more expensive. I
Ah,
think,
yeah.
too, with the cell therapies, I think it's one of those things where the toolbox isn't nearly as mature as it is for bispecifics, right? If you've got the targets, if you identify novel targets with bispecifics, you've got platforms, the knob and hole, whatever, to be able to get these bispecifics together and some multispecifics, whatnot. Whereas with the cell therapies right now, it's demonstrated utility in the hematological malignancies, right? We've got the anti-CD19s, the anti-BCMAs, hasn't demonstrated efficacy to this point in solid tumors. It's certainly working towards that way. And then we always talk about the difference between the autologous and the allogeneic, and wanting to get it off the shelf so that it costs less. And so moving along in that direction as well, but more to come. It's just not as advanced platform, let's say, as bispecific.
Absolutely. Yeah, and I am sort of chomping at the bit to get to our paper. So maybe as I'll finish up our investment news here. We talked about vaccines, we talked about BioNTech, and anytime you talk about vaccines and BioNTech, then we'd be very remiss not to at least talk about Moderna. So I've just got a general comparison between Moderna and BioNTech for our second sort of investment news that we mentioned. And here it really compares what BioNTech and what Moderna are as companies, like what their focus is.
Well, and we all remember these were the big companies for the COVID vaccine, the mRNA vaccines, and essentially coming out right about the same time. And those were the options, along with there was the J&J one at the outset of the pandemic, right? And so now, to your point, looking at the direction these two companies have gone and changed their portfolio. Whereas, BioNTech certainly has diversified into not only having the mRNA based cancer vaccines, but then doing antibody drug conjugates, CAR T combinations, so on and so forth. Whereas the Moderna has really doubled down on their mRNA platform.
That's it, right? And that's why it's, I guess, not as surprising that BioNTech is partnering with BMS on a bispecific antibody, right? So nothing to do with RNA at all. Whereas Moderna is working with a number of other pharma partners, I believe Merck, if I remember correctly, on their mRNA adjuvant vaccine for oncology. So both are trying to get into oncology, but Moderna has sort of a one type of molecule approach to it, whereas BioNTech has a multiple sort of platform molecule that they can use to target those sorts of things.
Yeah, absolutely.
Yeah. So interesting there. But yeah, Moderna's chugging along as well. Of course, neither Moderna or BioNTech are likely super happy with vaccine directions, but it seems like they will have other things that they can focus on there.
Now, speaking of chugging along, we've got this thing called MAFCA. Why don't we talk about that a little bit?
That's it. So in terms of KCAS news, the big thing for KCAS is that we did participate in the first ever MAFCA meeting. So MAFCA stands for Mid-Atlantic Flow Cytometry Association. And it's basically a flow user group here in the Philly area that's just dedicated to talking about flow.
Yeah, and so we had some fantastic presentations, and as well a panel discussing Cyto, which I got to participate on, and then happy hour at the end. I think really just making the connections with the industry, is one of the primary goals, and then from there learning, right? And certainly was able to learn a lot. I love the presentation. What was it by GSK on automation? That automation in flow. I think really is an avenue that we're keenly interested in, right?
Yeah, so it's one of those where it has information that's directly relevant to what we're trying to do, and it's a great spot where the local community, so everybody from our site was able to basically come out. Like we had, I think, 10, maybe more of our scientists there. Whereas we can't send all those people to Colorado. Our sponsors really frown on it if we tell them that we're not going to like just run samples for the next like 2 or 3 days while we all go learn things. Like they really don't like that. So having this where it's an afternoon where we can go and network with our peers and make sure that we're all aware of industry best practices and things like that, invaluable. So looking forward to more meetings with that group.
And to your point there too, I did let you know that no timelines were impacted negatively on this. Everything got done. So yes, no problems there.
Excellent, excellent. All right. Yeah, but as we said, chomping at the bit for this paper because it has all of our favorite things, right? It's got cell therapies, it's got immunotherapies, it's got autoimmune disease. It even talks about flow a little bit. So all these things are what we're looking for in terms of papers. It is a review paper, though, of course.
Yeah, and diving in, we had talked before about my thesis work, and I had done chimeric antigen receptor work in an alternative format, looking at macrophages, and so a lot of this is really near and dear to my heart because I was looking at ameliorating inflammatory conditions, autoimmunity type, specifically looking at RA at the time, and where is the need here? We can dive in. A lot of these types of conditions are maintained by immunosuppressive therapies. So think glucocorticoids, small molecule therapies, and then we've got biologics too. And so the aim of these are all to essentially rein in the immune system, prevent the immune system from attacking self. The problem with that is that long-term immunosuppression could leave the patient susceptible to disease. And so what a lot of these therapies are looking to do is to resolve the condition, right? When we think about when we take antibiotics, it's not just to reduce your fever. Like here, you could think about it too, is you've got cold medicine and it reduces your fever. It's not getting rid of the sickness. It makes you feel a little bit better. Whereas when you take the antibiotic, if you've got an infection, then that will make you better because it's going to kill the bacteria. And so here, these two types of treatments, the immunosuppressive therapies, the glucocorticoids, they'll make you feel better, but they're not changing the underlying condition. And so here we've got multiple different platforms that are aiming to change that. And I love how they're talking about the targeting too. Specifically, how with cancer, we're looking at anti-CD19 for CAR. And what does that do? That by and large destroys your B cell population. And it kills the cancer at the same time. Now, when you do that for an autoimmune condition, it has the potential for resetting the immune system, resetting that inflammatory condition. But it's very broad, right? And so what the aim of some of these other technologies they talk about here are making it more specific, using an autoantibody receptor, for example, and trying to hit just those B cells that are producing the antibody that is creating this condition.
That's right. Like think about it as tuning the PK, if you will, of a drug. So if you need just a little bit of a drug to get to that therapeutic effect, and then too much could be toxic, that's really what we're doing. But we have to do it by, I guess, targeting the right receptor as opposed to, changing the number of cells necessarily that are in the system. That being said, this paper did also go into the use of not just T cells, but also multiple types of cells.
Yeah, you got NK cells, you got Tregs, mesenchymal stem cells. It's so many different things, different technologies. This is where, we were just talking about is expanding the toolbox. And the toolbox is not as mature, right, as those bispecifics, but there's a lot of potential there. Really liked seeing the NK option, right, where there's just a difference in alternative cytokine release profile, as they had mentioned in the paper, meaning that there may be less likelihood of that cytokine storm, right, that has been problematic for the T cell chimeric antigen receptors that, we're all familiar with. Was it Emily Whitehead, and what was it, IL-6, and utilizing the anti-IL-6 to prevent cytokine storm there and save her. This offers an alternative that might not necessarily do that.
Exactly. And, as you say, as these things get more targeted as well, understanding what they're doing requires better and better flow cytometry. So again, this is what our tie-in here is, that, with the advancement of cell therapies, with the investigation of using them for autoimmunity, it's no longer sufficient to say, like, oh, do you have like B cells, or did you kill all your B cells? Now it's got to be, is this type of cell activated? To what degree is it activated? Is it functional? Is it excreting or secreting these types of molecules? Or is that activity reduced or controlled to the point that there's no longer a disease state?
Mm-hmm. Yeah, and I was just thinking about, for example, with cancer, you had radiation, which is targeted to a location, right? You have chemotherapy, which is very broad, right? The way that it works is specifically targeting the most rapidly dividing cells, which is generally cancer cells, but it also does kill healthy cells. And then you start moving to other therapies that become more and more targeted as we understand the disease mechanism, to be able to get to something that really only hits what you want, that smart weapon, and doesn't have those off-target effects. It's just that it's not a very easy thing to do. One of the cool ones that I saw here was ex vivo expansion of Tregs and then reinsertion. So, expanding the Tregs for 2 to 5 weeks. I did not realize that over 150 clinical trials have tried this. Is that something you had seen?
No, yeah. I mean, I did notice that in the paper, but now that you mention it, that does sound weird. Like, you think about the number of Treg therapy companies, and I can count them on one hand. So I guess maybe these are like smaller scale clinical trials to investigate feasibility elsewhere.
Yeah, perhaps.
Interesting. All right, I'm gonna have to look back into this now, Adam.
And so, to your point about why is flow cytometry important, right? When you are eliminating these cell types, you want to monitor it. You want to see what happens. A lot of the clients we have want to watch either the establishment of their CAR Ts, right? And so there may be a marker on the cell that they're using to identify these cells. And then at the same time, monitoring the target. And so looking for that depletion. And over the course of time, what happens, right? Do we continue to maintain those T cells? Do we continue to maintain the depletion of, example, the anti-CD19s, they have the B cells? And then what does the immune system, how does it respond, right? How is the patient responding? All of that information is very important to be able to understand. We talked about having reinfusions and if that's necessary, although in the podcast did a couple months ago, was talking about the reinfusion not necessarily providing much of a benefit, right? And so the understanding of the response here becomes a core in driving how the treatment might unfold.
That's it. And it's always an interesting thing about drugs that can't be redosed, or that there's not a therapeutic benefit of redosing. For these so-called one and done treatments, it's always, how do you pay for these things later on as well? It goes back a little bit into the financial realm, but, if you give someone an immunosuppressive and you say that person has to be on an immunosuppressive for the rest of their life, the cost of providing that could be, hundreds of thousands, if not like $1 million. So if you come with a cell therapy that's going to be quote unquote curative, do you have that person pay one time that the same cost they would have paid for the immunosuppressive and say like, you're better off now, so you should be happy with it? Or do you have them pay based on the cost of the cell therapy? Or do you have them pay based on the number of years that they get back that they live disease free?
See, this is where I would love to have that pharmaceutical financial person who works in this area kind of describe what is the calculus that goes through the pricing of it. And we've seen in the news that there's a policy change, most favorable nation status about the pricing of drugs, and try to balance out how much the US pays on therapies versus other countries. And, I don't want to dive into the politics of it, but at the end of the day, people should be able to have access to the therapy that would save them or would help them. And so at least there is discussion in that direction. I would just love to know more of what are all the factors that go into weighing the prices there.
Yeah. So in the pharma field, they usually call them the health economics outcome researchers. HEOR. So I'll try to find one that's either retired or willing to speak out and tell us like what sort of things go in there.
Sounds like a Netflix special, the way you just set that up.
Right?
So do we have to blur them out and change their voice, or like what's going on here?
They've been anonymized for their own protection. Yeah, indeed. But we'll find somebody that can give us a little bit more insight into that, and I think that'd be a very interesting discussion overall. So.
Yeah.
All right. And anything else that we want to really mention from this paper? Did we go over the impact factor?
So we did at the outset. You said we would have an impact factor of 7. The impact factor of this publication is 7.9, which is pretty high. So the publication is Seminars in Immunopathology. And so the paper, Current Advancements in Cellular Immunotherapy for Autoimmune Disease, and so was published just January of this year.
Okay. Excellent. And as you say, really walk through all the advancement in CAR-Ts and the different types of CAR-Ts and how people are trying to use it as more of an established tool set to then go after indications, so that way you're not limited by what the or the therapeutic in this case can do, but really by what you can imagine in terms of targeting modalities in the future.
Mm-hmm.
So,
absolutely.
But excellent paper and excellent discussion overall today. So, I guess we'll move into the wrap up section here and go to our fan feedback request.
So before we do, I wanted to go back to our MAFCA stuff.
Yeah.
And I'm going to put something out there. You and I discussed it a little bit. This became a bit of a discussion last night. And I think that I would really love to have somebody join us here to discuss reference controls, right? And it's something that right now the industry is trying to coalesce around for spectral flow cytometry, and would love to have a conversation there on best practices that different places use. I know how we approach reference controls, and I think that there's always a balance of how to do it properly versus, cost effect and best data and all of that. And so, I know that you didn't have this on the agenda, but it's just something that has really been sticking in my brain. So I'll just ask for forgiveness rather than having asked for permission to mention it on this.
Now, your presentation of this topic, Adam, was a little bit more civil than we were initially envisioning, right? Like, technically what we were talking about last night was we would like to host a cage match with a proponent of multiple different approaches to fight it out on the air here. So we will be looking for those people, and whether it's in the July episode or the August episode, I think that type of discussion will be good. And of course, everyone will be polite and things like that, and we'll be professional about it. But definitely there's some strong feelings about what the possibilities here are. and what they
You
really
sure?
mean for the baseline science. So we look forward to that discussion and agree that that amount of engagement would be great.
Yeah. And how are you going to have people be civil and have a cage match?
We'll see. We'll tailor that name, so it's got to be somewhere between a discussion on, which is the highfalutin version, and a cage match. So I'll try to come up with a good intermediate word that really shows that we expect this to be an animated discussion, and hopefully get to some interesting conclusions here.
I think you should call it the Summit on Reference Controls.
Okay, I like it.
Summit on the Mount. All right, so I completely diverted your point, and I know you really want to get to the next phase, so have at it.
Yeah, indeed. So the other fan feedback request, if anybody is able to comment on the podcast here and let us know what disease indication is the most difficult to analyze. This paper went through a lot of the autoimmune conditions. Some of them are relatively well behaved, but we always refer to AML samples as just the wild west of immunology, because no one knows exactly what you're going to get whenever you look at those. So.
Yeah, the heterogeneity, it's just mind-boggling at times. We got scientists here who've just had such tremendous experience with it, and so, handle it really well, but it doesn't mean that it's easy.
That's it. Yeah. And then in terms of things growing where they shouldn't, that brings us on into the banter section. And there I wanted to understand, what is your favorite type of yard or housework?
I'm here to talk about like weeding and stuff like that. And the thing that went into my head was, I am notorious with my lawnmower for just running stuff over. Now I got a push behind mower. It's not powered or anything like that. And I maintain it. I've had the same one now, I think probably close to a decade. And so do oil changes on it every year, sharpen the blades, change the air filter. And the thing is humming along still. But I have changed the blade a few times. And so I tend to not really do too much weeding. I just go over to the flower bed, and anything that is not a flower tends to disappear.
Along with a couple of flowers, maybe. It happens.
And so I got a Bluetooth headset. That was like one of the, what is it? Like a company birthday thing, right? We did that engagement committee thing for birthdays. And so I ended up getting the Bluetooth headset. And so now I like to listen to some music while I'm mowing. I will say, for those of us, I'm thankful that we're a podcast and we're not a TikTok video or anything. And I say that partly because I got this cut on my head. I'm the person who shaves his head every day. And I first used the Bluetooth headset, and I'm coming out of the shed with the lawnmower and did not gauge the difference in my height with the stupid headset on. It just smashed right into that stupid thing and got a big cut. Anyway, I survived, and I still mowed the lawn. Nothing was harmed but my pride, I think. So long way short, I don't know. I love being outside, right? I like to trim up the bushes, trim trees, mow the lawn. I hate weeding. That's why I use the lawnmower. The blunt force, I don't have a smart weapon there. How about you?
Yeah. Indeed. So I think for me, the best type of yard or housework is something that's going to propagate by itself in the future, right? So I absolutely love the fact that I've planted weeds, and the weeds happen to grow me food, and they can also compete out any other weeds that are growing around them. So blackberries in Georgia grow quite aggressively.
Really?
Oh, yeah. So if you plant some anywhere, they will eventually take over your yard. But they will also, again, give you berries and kill any other weeds that try to grow. So
that's interesting. I never thought of berries as a weed. Now, I remember going to botanical gardens when I was younger, and they were basically demonstrating that certain types of weeds could just be used in a salad. So we had a weed salad. And no, that is not like weed as in cannabis. This is a weed salad using weeds that are plants growing that normally you wouldn't want growing in your backyard. And so we had taken some. It was pretty good. I mean, it was green stuff that you put some salad dressing on, and salad dressing makes everything better, I guess.
With enough ranch, right? That's what my children think. So
I like vinegar myself.
Yeah. All right. Well, excellent. So, yeah, this has been a great episode. Thank you to all our listeners. And I believe the next one will be in July. So that is planned as the nitty-gritty of global harmonization.
Yeah, it sounds like fun. And keep an eye out, too, for your proposed cage match.
August for the cage match. Absolutely. So thanks, everyone, for listening. And we'll talk to you next time.
Thanks all. Take care.